We're happy to have this presentation. This is an opportunity to learn more about ClinGen and the tools that they offer researchers. Our title of this presentation is the Clinical Genome Resource from ClinGen. This is a community resource defining the clinical relevance of genes and variants. This presentation will give a broad overview of the Clinical Genome Resource and the ongoing curation efforts helping to define the relevance of genes and genetic variants for research diagnostics and precision medicine. I'd like to now introduce our presenter, Dr. Courtney Thaxton. Courtney is an associate professor and director of the ClinGen affiliated Biocuration and Coordination Core in the Department of Genetics at the University of North Carolina at Chapel Hill, where she leads a team of individuals in building frameworks, guidance, and specifications for assessing the strength of evidence for genes and genetic variants in disease. She also led efforts within ClinGen to develop and improve frameworks for gene disease validity and defining disease entities and engaging the broader community to contribute to ClinGen curation efforts. So with that introduction, I'm going to turn it back over to Dr. Thaxton.
Great. Thank you so much, Grant, for that wonderful introduction. And thank you, Latifa, for hosting this. I'm really excited to be here today and talk with you all about ClinGen. So I'll go ahead and start my screen. As we said, I do a lot of things in ClinGen. I am the current steering committee chair, and I hope to review for you Clinical Genomes Resource and how we've become a community resource defining clinical relevance in genes and variants for use in precision medicine and research. And so I like to start my talks with a little bit of an interactive element. So for those of you here, if you will humor me, you can go ahead and put in the chat if any of you feel confident enough to come off of mute and say something. I want to play a game called Let's Guess This Color. So if you all want to put in your idea of what you would call this color, I'd certainly appreciate it. Great, so I've got magenta, pink, mauve, purplish pink. We can have fuchsia we have in there. So several different colors, right? And so of course, people interpret colors differently, and that's based on our prior knowledge and our experience. It could even be based on our bias. And so really we have to ask who's accurate out of all of these? Who's inaccurate? And how do we actually tell, especially if this answer is going to inform treatment and life expectancy? As a color, it doesn't really matter, right? But when it comes to our health, it may. And just like we interpret colors differently, we can interpret information about genetic variants differently. What you're seeing here is a screenshot of a variant and the different classifications of pathogenicity that's published in ClinVar. And we can see that it ranges from pathogenic, saying this variant is causing disease, to benign and saying, I don't even know, a variant of uncertain significance. And if you were a provider coming with a variant in this individual, you may be very confused about who's right, who's wrong, who's accurate, who's inaccurate. And going through, you can see that there's different levels of information. So let's play this color again. But this time, everyone here, I'm going to show you the colors. And when I count to 3, and I go 3 and I say enter, I want you to enter in the color that you call this. So 1, 2, 3, enter. Great. So those of you who are listening afterwards in the chat, everyone said magenta here. We can go one step further and say it actually has an identification code. So no matter what name we call it, we will always know that this mixture of red and blue and greens or yellows is going to create this same color. That might be important later to this call as well. So if we think about everyone here and what we just did, what was different between your first and your second guess? And I think if we look at it, we're going to say it was data. I presented more data, or I shared more data with you. I also provided a framework or standards because I told you exactly what colors and what their names were. And because of that, what we can see is it did improve the answer for many people. Some got it correct, magenta. Others may have been something slightly different. But overall, as a group, when we came to share this data, we provided consistency across our approach because we used a standardized framework. We had similar data to assess. And so now we all came to a shared conclusion. And so really this was all to show you why ClinGen was developed in 2013. It was to create this authoritative public resource defining the relevance of genes and variants for use in precision medicine and research. And that's because as we came out of the Human Genome Project in 2000, sequencing became and coming into the clinical aspect to diagnose individuals. We knew there were several genes that could potentially cause diseases, and we wanted to know which variants were causing the disease. And so we'd have lots of interpretations, and some people had lots of data, other people had very minor data, or they wouldn't share. And so above and around all of that, we didn't have standardized frameworks. So it became a he said, she said, I said, they said type of approach. And so ClinGen was really established to be able to help go through these interpretations that are conflicting, providing frameworks that individuals can use out in the community to help us to have a shared understanding and obviously share data. It is funded by 3 core grants. You can see them listed here. We also have co-funding from other National Institutes of Health institutes and centers, one of them being the National Cancer Institute. And so the main goals of our consortium are to aggregate or collect and share relevant data about genes, genetic variants, and their conditions through the development of our infrastructure and standards. So we build several curation interfaces and systems that groups within ClinGen can use. We even have some that outside external individuals can use, like our variant curation interface. And it is a way for us to collect this data. We're reading the majority of which is publicly published information, right? Manuscripts, journals, but we can also collect data from individuals that are actually getting this from patients and individuals in their institutions, in their labs, etc. Then we go on to curate. We either develop or we update existing frameworks to make them more transparent, more usable, accessible to other individuals so that we can have a consistent approach on how we evaluate genes and genetic variants in disease. And this is all supported through our international expert curation ecosystem. Obviously, once we collect all this information, we don't want to keep it to ourselves. We can see that if we don't share data, we're going to have different interpretations. Whereas if we share data, we can come to a better, more consistent understanding of that gene and whether or not it causes disease as well as variant. So we do disseminate our work in an open access and sharing of evidence through our website. Our website is open to all. We also have data tools and frameworks that are available for anyone to use and download. And we also try to have our standards through continuous use of the FAIR principles, or findable, accessible, interoperable, and reusable. And at the effort of all of it is we want to continue to evaluate and improve our standards, our interfaces, and our frameworks, right? We need to define, develop, and implement these methods and metrics and enable the evaluation, and also to assess and provide improvements. We know that data is changing all the time, evidence, sequencing technologies, and we need to be able to adapt to those with our frameworks. And so our key goal is to curate the clinical genome. We know there's a finite number of genes that are associated with disease. We are particularly focused at the moment on monogenic disease, or a single gene disease entities. So that's just less than 5,000 genes. We also want to go through and make sure that we're looking at genes that may have actionability to assess whether or not there are actionable items, but also in that actionability for variant specifications. Certainly those genes that have the highest number of conflicting interpretations across different variant interpretation sites like ClinVar, LOVD, we may also focus for variant pathogenicity, and so on. And we do this by having an expert curation ecosystem. So the basis of all of our curation activities is bringing together groups and expert panels, which, as the name implies, is a group of individuals of experts that can both have clinical, genetic, as well as research and experimental expertise. We have biocurators, who are those individuals that are going to go out and read the paper, extract those data elements that we need, and be able to apply the frameworks and bring back to experts so we can weigh that evidence. And we can't do any of this without our coordination of our teams, right? Bringing together international members on meetings, being able to coordinate these curations when they're performed, when they are completed, is really essential. Through that, we have our frameworks, which occur through our working groups. And so this is where we develop standard operating procedures and update them. We provide training across all of our expert panels and all of our members because they need to understand these frameworks as well as the interfaces. And we also provide recommendations not only to ClinGen, but out to the community for what they need to consider when they're evaluating genes or genetic variants for their pathogenicity for disease. We couldn't do any of this without infrastructure. So we do have dedicated members and staff that go and build interfaces and databases, as well as any other resources that we need in order to complete this work. And finally, we want to disseminate this to all of you. And so we have a website that's open access and freely available to the public to be able to see and download the information that ClinGen is providing. And of course, as I mentioned several times, that this is a global effort. So currently, ClinGen has over 2,700 members. We are closely approaching 2,800. So if you have interest, we would like you to join, and across 72 countries and over 1,000 institutions. You can see that we have some gaps in gray in our map that we would love to fill with additional members within ClinGen, but we could not do this without this global community, and it is a very largely volunteer effort, and we are so appreciative that people want to come together to share data and be part of these teams to help improve patient care. Because of this resource and our global effort, we were recognized as the Global Core Biodata Resource in 2023, I believe. And so that's really a testament to the efforts that we are doing to make sure that we are building a resource that is usable by all. And so just as you see, this is a snapshot. If you were to go to clinicalgenome.org and in the search bar where it says gene search, you type your favorite gene. This is information you can access. You'll have any information on gene disease validity where you can click into the green box and see a full report of information. The same for dosage sensitivity when we think of haploinsufficiency or triplosensitivity, actionability, and variant pathogenicity. And we'll go through a little bit more of this later in detail. So we have 7 main curation activities at this point. We do have gene, variant, dosage, actionability, pharmacogenomics, somatic cancer, and a baseline annotation. I'll go through some of these in great detail, like gene and variant, because that is the most highly sought after kind of information from ClinGen, but I will go through some of these other ones as well. So gene disease clinical validity is really at the heart, asking the question, what is the strength of evidence that a gene is involved in disease? This is a really important question for us. And for all the other curation activities, this is where it starts. We have to evaluate if a gene is even involved in disease before we can think about if a variant is pathogenic, whether or not there's actionable items to take off of that, and so on and so forth. And so really want to stress the importance of understanding to begin with if a gene is involved in disease. So we've built a semi-quantitative framework that assesses the strength of evidence supporting a gene and a disease relationship. This looks at things like genetic evidence, including individual case reports, case controls, as well as segregation. And we also have experimental evidence that we bring into play, which can look at biochemical function. It can look at whether or not there are model organisms to support this, and if there are any types of rescue experiments. And so it really helps to define the critical need to demonstrate causality. We need to understand whether this gene is involved in a specific disease or with phenotypes and which phenotypes to describe. And it also helps us to organize and quantify different types of evidence so we can begin to regulate that strength of evidence in this points-based system. And it also allows our curators to methodically assign a clinical validity classification. You can see those classifications on the right. They range from definitive, which is saying, yes, this gene is absolutely involved in this particular disease or in these phenotypic features, all the way down to disputed and refuted. Nope, there is not enough information at this time, right? Positive or negative is disputed or refuted. We have published evidence to say to the contradictory, this gene is not involved in this disease. And so this becomes really important. We'll see further on just because of other authoritative bodies providing some recommendations based on these classifications. So it may seem pretty straightforward. Just whatever this gene has been asserted for a disease, just curate it, right? Do it. But what happens if you were to be presented with this information? You go and have a gene assigned to you, and you go look at a resource such as OMIM, the Online Mendelian Inheritance in Men, or you look in a Gene Reviews or any other paper, and you see that that gene is associated with 2, 5, 10, 15 different phenotypes or presumed diseases. Which one do you curate? How do you know? And this isn't a problem of just a very few. In fact, looking just a few days ago on OMIM, if you look on their statistic page, you can find that over 30% of the genes that are associated with monogenic disorders actually have 2 or more phenotypes associated with them. Well, this is an issue for us in ClinGen because the basis of all of our curations lie in the relevant data available. As we saw in the beginning, I gave you specific data. I could have given you data that wasn't in the context of this. So being able to understand what we include and what we exclude is really important because with those points in that semi-quantitative metric, that can change the classification and again, ultimately affect downstream patient care. And so when you see genes like this with MYBBC3, we need to be very careful because as we begin to split them, you can see the differences in the classification. And again, downstream recommendations will be affected. So what did ClinGen do? ClinGen did what it does best. It formed a working group and it said, let's start developing some guidance in which we can have individuals understand how they define the disease entity. What are the phenotypes included? What are excluded? How do we know what we should score those points? And that came in our ClinGen. We call it our lumping and splitting guidance. If you're interested, this is the Curated Disease Entity Working Group within ClinGen, and it's what Grant introduced me as part of an effort that I helped to lead. And so really this became a very essential element we had to do before gene curation after we had tried this a few times with that framework. We really needed to establish what is the disease entity. So we look at things like assertion, and we appreciate that there could be lots of assertions, but literature is biased. It's biased to novelty. And so what that means is that we kind of have to weigh that a little bit differently. We also want to more heavily weigh potentially molecular mechanism. Do we know that there are distinct mechanisms between diseases, things like loss of function, gain of function? Specific transcripts would help us to define that this could be 2 different diseases. But if we're seeing the same variant across multiple different phenotypes, and we're seeing families with those different phenotypes in the same family, that might be rather suggestive that it is a spectrum of disease, and we should call it that, include all the information. We also look at those phenotypic features, as I said, how do they segregate between families and within families, as well as inheritance pattern. And once we do this information, we call it a pre-curation, we also make sure that this is very explicit to you all and transparent. This is an example of a gene, MED12, that was associated at the time of the curation with 3 different phenotypes. It now has a 4th one. And what they found after doing this pre-curation is that these 3 seemingly asserted independent phenotypes were actually part of a whole spectrum of intellectual disability. So we ended up calling what we lumped it, we say it's a spectrum. We provided a new name. So we also have disease naming guidance for those of you that may be discovering new gene-disease relationships. And then we show you this information, and ultimately you can get to that definitive classification and know the phenotypes of individuals that are included in this. This becomes really important, as I said, when you go to variant curation, because you would be able to say any variant across individuals, no matter if they are diagnosed with these different features, could still be included when we look at the phenotyping code. And again, why does this all matter? Why does it matter for us to have these classifications? Why does it matter downstream? Well, one of the reasons it matters is because we now have the American College of Medical Genetics and Genomics has made formal recommendations for the use of these ClinGen gene disease validity recommendations for reporting genes for use in diagnostic panels. So what essentially this report says is that if you have anything from a moderate, strong, or definitive classification, and you have variations within an individual, you can return back results up to pathogenic. If you have limited, you cannot return anything above a VUS. So you can see how this would really change and shift the way we give back and report different variants. It may even affect which genes we include on different testing panels if they aren't actually part of that phenotype. You certainly wouldn't want to potentially keep including a disputed and especially not a refuted gene on a phenotype panel if the evidence does not actually shows to contradictory. It's not included. And so again, all of this starts to affect the diagnosis of patients and patient care. And what we hope is to be in a more positive approach. And so if you go to look now, and this may even have changed from yesterday evening till today, who knows, but always visit clinicalgenome.org to be able to see the latest. But we have curated 3,156 gene disease curations, over 2,608 genes. So we're halfway to that goal of curating all the genes associated with monogenic disease. And this is currently over 47 active gene curation expert panels. These span multiple different clinical domains, including cardiology. We have cancer, neurological disorders. We've got ocular, hearing loss, skeletal, and an array of them. So I definitely encourage you to go to the website if you want to see those domains and know what's being looked at across the genes for our different gene curation expert panels, or GCEPs. So from gene curation, we'll move into variant pathogenicity, because now we've decided what genes actually cause disease. Now we need to think about what variations within that gene are pathogenic or disease causing, because not all of them may be. And so again, in those clinical interpretations of variants, just like we saw in the beginning, ClinVar is a place where we can have many different interpretations or classifications for a variant's pathogenicity that are reported across multiple institutions. And sometimes those can align very nicely and they can give you the same. We have several of them, like you can see here, and there's thousands that actually have conflicting interpretations. And so again, what happens when these interpretations differ? And how do you know, as an individual coming here, a clinician diagnosing, which one of these is the trusted source? Some of your trust may come based on what more information they give. And when it's blank, then what do you do? And so this is where ClinGen came in. We decided that we wanted to have our variant curation expert panels, or VSEPs, to really work based off the ACMG guidance from Richards et al. 2015. Which nicely outline different types of data elements that they want to look at for variant interpretation and classification, and look at the strengths of those evidence. And so our variant curation expert panels go and use this as a basis, and then they look at that gene-disease relationship and they provide adaptations that are more specific. One example here that you all may know of is BA1, or the standalone benign criteria. Per ACMG's 2015 guidance, says if it's above 5% in the general population, it's benign. But if you have a rare intellectual disability syndrome that's seen 1 to 1 million, or even 1 to 100,000, you'll never see 1 to 5%. You need to have a different BA1 standard. And so that's what these groups go through and begin to specify. Our variant curation expert panels do have a 4-step approach in order to get to these specifications and release out to you all, the public. It does start with just defining that variant curation expert panel and their scope, what genes, what diseases they're going to work on, and make sure that we have the appropriate expertise within that group. They then go on and draft those specifications, the adaptations to that Richards et al. 2015 and ACMG guidance. They then submit that to what we call a sequence variant interpretation working group, but that is moving to be called the variant classification working group in the future. And then, of course, once you make those adaptations, you really need to pilot this information. So you need to take variants and look at a before and after those adaptations to make sure, are the classifications aligned? Are they improving, or not even improving, but being more accurate representation of the information that you have there, or making sure you haven't done something so detrimental that you need to go back and adjust those specifications. Once they're approved, they go out to sustained curation with the priority to look at those conflicting interpretations and try to work with those different institutions to be able to update those variants. All of this work that has been done for the specifications can be accessed through ClinGen's criteria specification registry. This is something that is available to everyone, and I'm happy to send a PDF afterwards of these presentation slides so you all have links to these to Latifa to send out. But it's a really great resource if you want to go into this criteria specification registry, or we call the C-spec. You can look up your different genes. You can download by clicking the button. You can see where they are. Released means that they're already at step 4. You can access them. Classification rules submitted means they're in step 2. So you can see where all of these specifications for genes are in that 4-step process. And of course, those again that have it released, you can access and be able to use them. And so, of course, the really important part of this is that expert panels combine data types to classify these variants. So when we have all of the optimal variant types that we need, or data types, we can end up getting to something like pathogenic. But if we were missing any parts of these, we end up at a VUS. And so this is really important, and why I started this talk with the game of let's play this color, because data sharing is at the most highest point of our priorities. We want to be able to get access to and give access to additional data across individuals so we can improve those interpretations. As ClinGen is working, as we said, we publish all of the information out to be as transparent as possible. But certainly being able to get more data from resources like you all have at GGMC would be really wonderful, especially whether it's participating on expert panels, providing data that you can share to us. We also know that you can have patients that share data with us through our ClinGen's Genome Connect. So this is ClinGen's patient registry, and patients can opt to share that genetic information, which is a really fabulous resource. So again, this is the patient portal. All the variants that are deposited or given here and shared are deposited in ClinVar. So it makes this information accessible not only for ClinGen expert panels, but for anyone who uses ClinVar and interprets variants. So really, if you have any interest to this, you can put in clinicalgenome.org/genome connect, or you can even just search Genome Connect in Google and it should get you to these pages here. And so it does seem like a lot of work that we did, this 4-step process. It seems very involved, but with good reason. Our process is FDA recognized. It's one of only 2 FDA recognized databases and frameworks for training and protocols for variant classification. And so this really starts to highlight the rigor and the consistency and transparency that we provide the community internationally about variants associated with disease. When you go to ClinVar, which is where we deposit all of our variant classifications, you can see those are FDA recognized. We'll highlight those from ClinGen's variant curation expert panels. You'll see extended information as well as being able to access more transparent information than what you typically would. One of those is our evidence repository. As part of our FDA recognition, we did have to build this evidence repository as something that could parallel and give more information than our ClinVar submissions do. So you can visit our eRepo and be able to look across either the genes that are already step 4 approved or by the expert panel and see the number of variants that have been classified, and you'll see them and can even choose based on their classification, again, from pathogenic all the way down to benign. If you were to click into one of these variants, you're going to see information of our evidence summary, which is also equally published on ClinVar, but you're going to see on the evidence repository each particular code that was met and the additional information that we bring about that, including PMIDs, whether or not that for variant curation, I will say we can use unpublished data. So we will say this is unpublished data that allowed us to get to this. We are very careful about HIPAA, PHI, but this is one area where we know it's really important to be able to share unpublished data to get these variant classifications out and help the community. To help to support all of this as well, we have our ClinGen allele registry. This is one area where you can register an allele to have a variant ID, a stable ID, which is really great. We know that names change over time. You can see that here. The ClinGen allele registry will bring that variant in. It will show you what that transcript is across all of the builds that we have so far. It will show you also between the ensemble and NCBI, and it will indicate what is the main select, and we're working on main clinical. You will get that all important ID here so that, again, if it is referred to differently across different papers, we know we're getting to the same variant and we're not duplicating efforts or leaving out any information. And we bring in some really nice handy links if you want to go look at additional information like Nomad, Exact. We bring in PMIDs from ClinVar, etc. As you can see here, we are approaching 3 billion registered variants, and that was as May 2025. And you do have the ability as a user to bulk upload and acquire and register variants. It is API accessible. And so again, this is open for all to use if this is something you would find to be very beneficial within your work and institution, and start implementing the use of stable IDs so that you know you're talking about the same variant. Please consider looking at our ClinGen allele registry. So far, and again, this was current as of last night, we have gone through almost 11,000 variant pathogenicity curations. We have 68 active variant curation expert panels. Oh, sorry, this is supposed to be a VSEP down here. We have 5 more applying, but I would say across the 68, this is maybe 28 of them that are at this step 4 process. So we do have some that are still working through the process, and we plan to continue to increase the number of variant curations that we have across multiple genes. So for clinical actionability, we really start to look at whether or not there are actions that could be taken to improve outcomes for patients with a genetic risk. And so that overarching goal for their curation process is to identify human genes that, when significantly altered, can confer high risk of serious disease that could be prevented or mitigated if the risk were known. So equally like gene curation and some other curation activities, we do have a semi-quantitative scoring metric for that to assess the outcome intervention pairs. This will be based on things like severity of the outcome, likelihood of disease, effectiveness of that intervention, and the burden and risk to the patient. This information can be used to guide decisions on reporting secondary findings, and this is really where this guidance is especially useful. The clinical actionability curation group has 4 subgroups. You can see them listed here. They focus on adult and pediatric actionability, and the framework that both of those use is on the right, and you can see the PMID if you want to see the latest paper on this framework. They also have a polygenic risk score clinical utility working group that is coming up with recommendations and guidance, publishing about the use of PRS for actionability. And the newest group that they have started is the population screening working group. And again, building a framework here to look at different population screening and actionability. So thus far, we have 498 total gene disease pairs that have been evaluated for clinical actionability. You can access any of these reports from clinicalgenome.org. And I know these are things that are part of the United States ACT sheets that they use as part of ACMG as well. So if you are looking at having more information about what you should report back for secondary findings, please take a look at our actionability working group. So one of our other main activities is dosage sensitivity. It's one of our first curation activities that started, and it really assesses whether or not loss or gain of a copy of a gene or genomic region can result in disease. So unlike gene disease validity, where we can take in many different aspects of it, including specific variations, as well as having haploinsufficiency, this is really looking at loss of one or a gain of another copy of that gene. They do use a scoring metric based on observations in human cases. So again, semi-quantitative metric, and the information can be used to aid in the classification of CNVs. This is part of the ACMG and ClinGen technical standards for constitutional CNV evaluation, and it can also guide that discrepancy resolution efforts between laboratories. So they do a lot of work to be able to have discrepancy resolution. So you can again visit the dosage sensitivity group on clinicalgenome.org. They have 3 subgroups currently, and they are building a new one. They do look at general gene dosage sensitivity. They've looked at recurrent CNVs, and they've had a hereditary cancer dosage group. In the past, they've also had one, I believe, on neurodevelopmental disorders. They also are evaluating biallelic loss of function as a part of the dosage sensitivity. So it's a really new, interesting aspect that's coming into play for this specific curation activity. Across the years on dosage, and as of yesterday, they have over 4,000 total dosage sensitivity curations across over 1,600 genes. So again, if you're really interested to see about the scores and how that might help you in diagnostics and everything, please check out dosage sensitivity reports on clinicalgenome.org. And I'll go through lastly somatic cancer. This again is looking at whether or not variation in this gene can cause oncogenicity. So it's looking very specifically in cancer and looking at that variant interpretation. This is part of our efforts. They have 3 different task teams. These are much like ClinGen's clinical domain working groups that are looking at their pediatric hematologic and solid tumor sections. You can see that there are several different what we call somatic cancer variant curation expert panels, or SCV sets, and they are in multiple different stages, and we are about to have several of them get to the point of being in step 4 in implementation. They will be publishing all of their information about their interpretations to CIVIC, which is the Clinical Interpretation of Variants in Cancer database. Those will also be published to ClinVar, much like our variant curation expert panels published for monogenic disease. And again, you can see all of the different focus for these SCV sets, including FGFR, EGF, established significance, and NTRK, and so on. We do have a few other activities that I won't go over here today just because of time, but I do encourage you to look at them. We have a pharmacogenomics group where we're bringing in information from PharmGKB and CPEC to look at the differences of how genes and their effects are on drug response and vice versa. So if you're very interested in pharmacogenomics, I encourage you to go look across the ClinGen website. We also have an activity that's very close to my heart called baseline annotation. And this was a way for us to be able to engage the community, regardless of what your specific expertise is, and reading and annotating the literature in a standardized way with those all-important variant IDs and other IDs so that we can be able to assist ClinGen expert panels in finding variations for their work. And so again, as Grant had said in the beginning, I also do a lot of work in engaging the community. I'm one of the co-chairs of the ClinGen Community Curation Working Group. And so if any of you have been wondering as you're here or listening later on online, how can you get involved? We do have the C3 working group that was established in 2018 with the mission to engage, identify, and communicate with potential ClinGen volunteers and to be able to assist their placement. And our motto really is, from clinical geneticists to citizen scientists, everyone can contribute and anyone can curate. Obviously, there are caveats there because you have to have a computer and be able to read and type, but we really feel that we want to make everyone feel that they can contribute to this resource. And I can say we have had citizen scientists with no background doing baseline annotation and really helping to find papers of interest for groups. You can see that we have now over 2,700 respondents to our survey as of April, and it's always growing. And we've actually reached out to 96 countries. So you may say, wait a minute, you told me there's 72 countries in ClinGen. Well, these are people we're still waiting to train to get involved. Sometimes people may or may not come into play, but we would love to get more people into ClinGen in our expert panels for this effort. And so what can you do? We certainly have 2 different activities. If you go to the clinicalgenome.org homepage, you can see that you can either volunteer as an expert and take a survey. We will ask for some information about your expertise background and where you would like to participate. And if you would like to volunteer to be a biocurator and be able to collect information, you can do that as well in our volunteer to curate, which will take you to our C3 survey. And I just want to thank you all for your time and take any questions. Certainly, if you would like, we have a QR code to take a survey. We do hope to have a follow-up workshop or 2, and there are 3 different choices for you all to think about for this particular follow-up workshop. And if I can get to, I'll end my slideshow. I'll also put this link into the chat for you all.
All right. Thank you very much, Courtney. That was absolutely fabulous. I've been aware of ClinGen for many years now. However, have not seen a presentation about everything that ClinGen is doing for several years, and what I just saw is amazing. The
Thank you.
extent of what ClinGen is doing is just quite very impressive. I realized after Courtney was began her presentation that I failed in a major area of being a moderator, and I should have said in advance that if you had questions for Courtney, to put them in the chat. I think also at the same time, if there are um, if you have a question and you just want to raise your hand, then we could do that too. Is there anybody that has a question immediately? I've got a few that I can ask, but I just want to see with those that are on the call right now. Does anybody have some questions for Courtney?
I have a lot, so I start with one. Um, I think it's going to be a hard one to answer, but in general, if you can give like an overview statistics of how many of variants get upgraded from unknown to pathogenicity, because my hunch or knowledge is like that we're over the 90% that variants get downgraded.
I don't have that particular statistic. It's a good one. I can certainly try to find out. We do have several groups that begin to look at how we change those. But I think you're right. In some respects, there are some variants that may go into a VUS category because we may be more conservative of our approach, right? Asking for more evidence. That might also be because of the way the ACMG 2015 guidance is currently, right? Everything starts as a VUS. They are updating this, which many of you may be aware of, where there is going to be what they call the SVCV4 or the sequent variant classification version 4. And at that point, everything's going to start as likely benign. So they're going to be innocent until proven guilty, right? And I think that's a way for us to try to get past the number of VUS that we continue to have in ClinVar, right? Which is over half of the variants that are reported there. So I think we have been moving some more into the likely benign or likely path area, but I couldn't tell you exactly the percentage. I'm sorry.
Okay, and just another question. How, at least in the States, are labs obliged by law to do these reassessments periodically? Or once they give out the first result, it's always the clinician's responsibility to
that is a good question.
review what is the legal
Yeah.
standard in it. I know that
I'm
it's treated as this on a regular basis.
right. I'm not sure that there's any legal standard that says you have to do this at this point. Certainly there, and if anyone else knows better than me, please speak up. I do know that a lot of, as you were saying, diagnostic companies will reevaluate. Certainly there are other institutions that will reevaluate. I think it also can become difficult based on the fact that patients can change states, institutions, other places. And so they get lost to follow up, right? And that could go into a whole nother thing of us talking about shared data, right? Going into shared medical records and, Epic and other things. But I don't know that there's an exact law that they have to do it. It would be great if things are moving to reassess. And certainly that would be a very effective for precision medicine and health, right? Is like as things come up to play and we have more indications. Now, ClinGen's Genome Connect, I can tell you for that patient registry, they do reassess and they go back and ask based on reassessments and tell patients, hey, there's been a new update to this information.
Uh, If I can speak, my name is Shubit Barua. I am a lab geneticist, and I can comment on that. There is no law, but there is a guideline recommendation from ACMG where they say that periodically variant or the gene can be revisited. But efforts is kind of joint call between the lab and the clinicians and even the patient. So if a clinician requested, lab have to do it. Lab must do it. And if there is any change from the lab point of view in a bioinformatics pipeline, or if major database has been changed, then the lab take a self-initiative to revisit those. And if there is any upgrade, they will do it. In general, it is said that around, if I'm not wrong, around 29 months is the right time when a reanalysis is requested, because it also gives that lag period when the patient has developed additional phenotype. Let's say it's a baby and it's now developing additional phenotype, which can be correlated with the variants, or certain new gene has been discovered, like ClinGen gene discovery has associated certain gene with certain new syndrome, which now fits with the patient's phenotype. So it is a kind of, there is no law, but it is a kind of, whether the lab has a particular SOP and whether the clinician has a kind of inclination to revisit within that time frame. But it also brings a lot of burden to the lab, because it also takes a lot of time, effort. So there are a lot of things goes on, how the CPT code and how it will be built and how far the lab is flexible. So most of the lab, for the first time, when they're reanalyzing, they do it for free. But following that, there is a CPT code which has been inserted there. So it's a kind of no kind of flat guidelines or recommendations, but law, but there are certain recommendations how people can do. Thanks.
Thank you.
Excellent answer. Thank you. Anybody else have a question? You don't have to raise your hand. You could just speak up.
Yeah, Grant, Gabriela has a question.
Okay,
Please
good.
around.
Thank you, Courtney. This was great. I don't know why my name has been expanded here, and I think there are three Gabriela Robertos on the screen. So I wanted to ask you about the response to your efforts to lumping and splitting these syndromes that have been associated with multiple genes. Do you have a sense of how the clinical community is doing with this? Has this been included in OMIM or gene reviews, which are other sources that us clinicians use a lot?
Yeah, very great question. And so certainly OMIM is going to do what OMIM wants to do, but OMIM is a representative on our curated disease entity working group in lumping and splitting papers. And so they do still connect with ClinGen and say when we have some differences, and their purview is different, right? Their purview is to say this is what was asserted, and it's really us that's coming through those frameworks. As far as clinicians go, we do have to have a balance. It is an effort of an evolution, right, of what we've been used to for millennia, right? Let's split things, let's move things out. And so really when we started to get into sequencing and getting to the process of pleiotropy and heterogeneity, right, this really became that complicated effort. So while I think a lot of people are adapting and moving to this because we can see, again, and where we bring them to it is, if you keep splitting, you have to exclude this data. Your classification isn't getting where you want it, right? And you can't put this in this variant interpretation. So I think that understanding is coming into play. What we just have to remind individuals is that even if we call something like that Med-12 related intellectual disability, right? I'll try to go up to that slide. I can find it. We're not asking clinicians to diagnose anyone with this name. They don't have to, right? You diagnose them based on their presentation when you saw them at the moment. I can go through several of these in cardiomyopathy where we're lumping hypertrophic cardiomyopathy with dilated cardiomyopathy with arrhythmias, right? And they are part of the spectrum, but you're seeing someone based on how they come to you, and that's where you're giving them that phenotypic diagnosis. We just want it on the level of the curated disease entity so you all can understand. Now, gene reviews is also kind of represented here because Les Biesecker is on this group and he's been part of gene reviews. Interestingly, I think they talk about these syndromes. The other part is gene reviews has a different naming that we have because they will talk about a gene that is associated with multiple names. So while they may call it Med-12 related disorder, we want to be more specific in our dyadic approach that it's a gene with a phenotype dyadic approach. So we think that it's moving. It's always a process, right? When we evolve, if we have pushback, but we try to work with our community and make sure that we're addressing everything. Now, one group that we do work with really closely is Mondo, the disease ontology. And so we're able to create these Mondo terms. We're able to change and update ontologies to make them more accurate. And that is one part of what ClinGen's working with. We appreciate what Mondo's done, and it was computationally evolved, and they certainly want experts to come in to help with these ontologies to make them more clinically relevant. And so that is an effort that we work with them to make sure those are represented. Oh, yes.
Courtney, I'm going to ask two of my questions. The first one is just I'm curious about the users of the tools that you have, specifically between research focused users and clinical users. Like this seems like an obvious tool for or a research user, but do you find like clinicians using this tool? I mean, will they take time in their practicing patients to use your tool? What
Right.
what is your experience there?
We do find that clinicians use this, but we do have an element where we're trying to reach out even more, right, to individuals who aren't geneticists and saying, do you know of ClinGen? Would you use ClinGen? What are you using most? I will say we do get more feedback of OMIM because, again, that's been a standard that's been used for over 50 years. But I think this is one area we're trying to get more individuals, but we certainly hear feedback that clinicians find this valuable and useful. Obviously, those molecular geneticists, genetics labs, right? They're bringing it in and even research. But I think they do find it, especially when they're wanting to learn a little bit more about a gene, right? Or they want to learn about what an expert panel is doing. What's nice on our website is that you can go see all of the genes that an expert panel has curated and think about like, oh, wow, here are some potential additional genes that cause that phenotype. Maybe I should think about my genetic testing panel I'm about to give, right, as a recommendation for a patient.
Do you find that most clinicians are aware of the tools that ClinGen offers them, or is that still a minority of clinicians? And is there opportunities to
Yeah.
sort of educate that
Yeah.
group?
We're always looking for opportunities to educate more people. As much as we try, we don't know if we're reaching everyone, right? And how well we're reaching it. We definitely see our impact a little bit more in papers and information, but certainly this is an area we would love the opportunity to hear more from clinicians and how our different resources may help them in their everyday kind of work.
So what I found is that at least the clinical geneticists are very well aware of ClinGen. Sorry. If not the general clinicians, and that bridge has been very much effective because genetic counselor are very aware of this. So they bring that bridge between the clinicians and the lab regarding the ClinGen and the effective tools, because I am also involved in ClinGen, certain one of the group, and it is led by a clinicians, but it is a clinical geneticist, a particular syndrome specialist like that. So a lot of clinicians are involved, but I don't think in general all the clinicians are maybe well aware or visiting, but clinical geneticists are really visiting very frequent in this tool.
And I'll take my question just one step further. You know, my background is healthcare delivery and building computer-based tools for those clinicians to use. Are you aware, Courtney, of any third-party commercial tools or even EHR systems that are accessing your data and incorporating your information in their tool? Because those are the tools that are going to be really clinician facing, right?
Right.
So clinicians would adopt it quicker if it's already in a tool that they're already using.
Right.
Yeah.
And certainly we've had efforts in ClinGen for EHR, electronic health records, and getting ClinGen guidance into them. So I think there has been efforts, maybe not necessarily like a ClinGen working group anymore, but amongst the principal investigators of ClinGen and different efforts to work with EHR companies like Epic and others to be able to say, can we bring in, when you have a genetic diagnostic report, a link to ClinGen and that gene, right? So that you can now have the access to see, have we looked at that gene disease validity? What are variants that may already be attributed? And then even linking it, okay, hey, look, here's the actual variant report. We do have a lot of efforts in trying to just get people, as you saw, the ClinGen allele registry, just to get them to register and use a stable variant ID, because then we can share data across different systems and, you know, whether it's EHR or somebody else's health system, if it's an industry partner who has their own database, with those standardized right IDs, we can share that data. That's also why we're part of GA4GH, right? The Genomic Alliance for Global
Genomics and
Alliance,
health.
excuse me, for Genomics and Health. Got to get all my G straight there. Because we really feel that having that standardized approach for data is going to help us to share it, right, more easily to many different parties.
Yeah. I was just as I was reviewing everything that you guys do, I was thinking to myself, wow, this is data and information that should belong in every EHR.
Yeah.
So that the doctor, the doctor doesn't have to take it upon themselves to develop a new skill set and go out and learn a new tool, or, leave the HR and go into another app, to do
Yeah.
that work. It should be integrated within the
Yeah.
HR.
And that's where we need you all as a community too, to speak up and say, wait a minute,
Yeah.
guess what? We want this, right? Sometimes it takes the community to tell those third-party vendors what you really want.
Yeah.
It's the same for us, again, back to those variant IDs. If we could just get people to even publish their information. So if you all here are publishing papers, if you would consider using a stable variant ID, that is going to make our life so much easier with AI, LLMs, anything, to go and find that data very quickly on that variant and be able to assess it, because that is the area we spend the most time, right? Is searching
Yeah.
for that literature and what's applicable. So having standardization is going to help across the board.
Courtney, we just have 1 minute left, and I'll just ask this one last question. I was just curious about the options for patients to upload their data. How's that gone so far? And is that an area that you hope to expand on?
Yeah. I think it's gone pretty well so far with our Genome Connect. And of course, we're always looking to expand. We want to get more individuals participating with this, and they've even gone so far as to make sure that they have translation. So they've now moved from English to also Spanish translation, and looking to do more languages in the future to make patients feel more comfortable, because they are having to indicate they're willing to share this data, right? And understand what data they are actually sharing. So I definitely think we are in this area that we'd like to improve and add more, but I can take you, find it real quickly. I'll share and I'll put it in the link to the chat if you all bear with me 1 second. So the Genome Connect on ClinVar can go and tell you how many variants we've already entered. So you can see it's almost 2,700 variants across multiple different genes. And these are reflective of patients who have decided to share their data. So it's there for you all to use, but certainly it would be great if anyone else wants to share their data in this part of this resource. We are happy to have them.
So I just sent in my saliva sample for all of us. All of us as
Yeah.
asking people to send in samples. When they complete that sequencing process, and I think I should have access to my data, that I can then upload it with your program.
Yes, I think so. And I don't know if they're looking more for individuals that are, diagnosed with something and having that kind of information, but you should be able to at least reach out to Genome Connect and see what you can do and what you can share.
Okay. Finally, Ricky will be our last. She has one last question,
course.
and then we'll end the session.
Thanks. So can we still say that functional analysis is the gold standard, or the, where we would like to be with every variant? Do you see a future when we won't have to have all these curations and really count on that? Because I put in the chat something that shocked me with the APC group, that that even functional analysis is not going to resolve most of missense variants for that specific gene. And that was kind of shocking to me because maybe I'm still
Interesting.
hanging on to old conceptions, but
I think for a majority of genes, and again, this is personally, that functional evidence is going to help. But just like we went through the process of how do you define that disease entity, if you have a gene associated with multiple phenotypes, your assay really has to be specified for that mechanism for disease, right? Otherwise, it won't be helpful because you could be seeing something and you don't know if it's just an artifact of that, massively parallel array that you have, if it's the cells. So there is specificity that has to happen. But I think if you get into really understanding the mechanism of that gene with that disease, then functional assays for that should be a very strong strength to tell you whether or not that is going to have an effect, that variation will have an effect in the area that you would find it. I don't know if we'll ever get to a time, it would be nice, but I don't know if that time is when we all are sequenced regularly and we have lots of genetic information at our fingertips and we can begin to watch someone's history, right? Like from birth to death, that precision health. And then as we're starting to see symptoms appear at different times, that there's just enough accumulated information, right? That we alone on a number of individuals that have that variant and those features can now say, yes, right, this is pathogenic. But I don't know, I guess I don't want to be out of a job as a biocurator, that I'd still like to think we would need all of this additional research, right? And things are changing all the time. Even if we figure out these monogenic disorders, are you really telling me only 5,000 out of 20,000 in our genome, right? We know that there are different things like RNA happening or somatic variation that's happening and occurring in the brain in specific cells. So we still have a long way to go research-wise, right, to figure out how can we access those cells? How can we, right, sequence them now? How can we do that without hurting a patient, right? And how can we find out more information and then going on to treatment? So I think we still have ways to go, and hopefully we're not putting anyone out of a job, but I do think collectively us all working together, sharing that data out and publishing our ideas really helps us to move forward.
Fabulous. Thank you. Thank you, Courtney, for an amazing presentation. And hopefully this will inspire people to get involved with ClinGen. Latifa,
Yes, please.
I'll turn it back to you.
Yeah, thank you so much, Courtney, for this is very insightful presentation. And also I would like to thank everyone for their participation. We'll be sending the recording for today's session so they can share it with their colleague who could not attend it. And of course, you shared with them the survey link, so to share with you the topics of their priority, and then we'll take it from there. So thank you again, everyone, and enjoy the rest of the day.
All right.
Thank you all as
very
well.
much.
Bye.
Very
Bye-bye.